Okay. Hello, everyone. Good afternoon, good morning, good evening, depending on where you are in the world. I'm seeing a lot of familiar names joining currently. Some met for the first time at GEM last week. Thank you so much to all of you for joining us, and welcome to yet another edition of the GSU Malaria Journal Club monthly series, which means we organize it on a monthly basis. Today's webinar is, interestingly, both vector and parasite focused, referencing Clara's recent paper titled Evaluating Evidence for Co-Geography in the Anopheles Plasmodium Host Parasite System. My name is Pablo Chauke, for those of you that do not know me, and I'm a training coordinator at Malaria Gen GSU, working with a team of awesome people to organize these monthly events. Those include Sri, John, Vicky, Christina, Alistair, Chris, as well as others. So there's a team of us behind this, and it's exciting to always host these journal clubs. Just a few ground rules for you attendees and participants. The journal club is being recorded and will be available on our Malaria Gen YouTube channel later today. So you'll be able to share the recording with those that couldn't attend, your networks, your friends, your colleagues, and for you to also rewatch the amazing and educational journal club that we're going to have today with Clara. Kindly stay on mute so that we don't disturb our speaker today. We will have a Q&A session at the end where you can raise your hand, where we'll allow you to unmute and speak to ask your question, or you can type your question or comments in the Q&A or chat box, and I'll read them for our speaker later as well during the Q&A. Without further ado, I would like to introduce our speaker for today, Clara Rehmann, a PhD candidate all the way in the United States of America, who kindly agreed to give this webinar when I invited her. And I had the pleasure to meet Clara face-to-face last week at the Genomic Epidemiology of Malaria conference in the UK. What a smart, authentic, and kind scientist. Yeah, that's what I'll say. And the title of the talk is, as you can see on the screen, which I find interesting, Location, 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 Evaluating Evidence for Co-Geography in the Anopheles Plasmodium Host Parasite System. Short bio to introduce Clara. Clara is a rising fourth-year PhD candidate in the Ken Ralph Co-Lab at the University of Oregon in the US. Her thesis is on the dynamics of positive selection in population structures across continuous space. In her research, Clara uses computational models and methods to understand how evolutionary processes impact patterns of genetic diversity. She's particularly interested in applying these approaches to infectious diseases and host parasite systems. Clara, thank you so much for joining us. You may begin your presentation.
Awesome. Thanks so much for having me. Thanks, Pablo. Great introduction. Yeah, it was really lovely to meet some of y'all at GEM last week. See some familiar faces. Yeah, and thanks so much for being here. I guess I'll just roll in, get started. Yeah, so I'll be talking about this paper involving how we're looking at co-geography patterns between Anopheles and Plasmodium using machine learning. And so let's see, can I advance my slides? Sweet. So I don't think I need to convince anybody that malaria is kind of an important system. And it's also a pretty dynamic system because we have three main players, the parasite, the vector, and of course us. And so we know a fair bit about how humans and Plasmodium are interacting with each other, as well as how humans and mosquitoes interact, in particular through like preventative measures like pesticides. However, when it comes to the dynamics between like the parasite and the vector, that is still kind of a black box. And it's important to understand kind of like the whole picture of all three of these organisms to get the best idea of what we can do for global health. So with that like little bit of background, I'll go into the outline of my talk. So today I'm going to be talking about what we can use, what we can learn about this system with supervised machine learning. I'll go into a little bit about what we can do with supervised machine learning, in particular, explain that process. then kind of discuss what we can do to detect patterns of cogeography and the different ways that we can interpret the errors that we're seeing in our machine learning results. Talk a bit about dealing with spatially biased data sets and how those impact our interpretations of our results. And then finally discuss interpreting our results around Anopheles and Plasmodium. So I'm just going to start out really quick with talking a little bit about supervised machine learning. And machine learning is something that we probably all have heard a lot about. I'm sorry, I lost my presenter notes. Excuse me. Hold on. I am very sorry. Um, there we go. Okay, apologies. So supervised machine learning. Um, this method, we start with a training data set and associated labels. And the goal of what we're doing is showing the network training data. The network sees the input data and will predict labels. And the goal is to associate the training data with true labels. The network does this by comparing predictions to the true labels, and then we update the network via something called the loss function. And so our overall goal when we're building this network is that we can show the trained network new data, and it will be able to predict labels for that data based on the data that it's seen previously. So as like a toy example here, we can think about image classification. So if I have images of lizards and images of mosquitoes with labels, we can show those images to the network. The network will predict labels for these images, will compare those to the real labels, and ultimately the network kind of learns what a lizard looks like, what a mosquito looks like, and we can show it a new picture and it will be able to tell us what that is. So this is image classification, not super biological, but you can imagine that this training data instead of images could be genetic sequences that we could associate with species labels. Um, to go kind of like further, you could imagine that these labels could describe evolutionary scenarios. Say you have genotypes from neutral genealogies and genotypes from genealogies under selection. Um, and these labels don't have to be limited to just, classes. We can have continuous labels like inferring a selection coefficient, for example. And this kind of method, training on genotypes and then some sort of label, is really useful in population genetics. It's something that my lab focuses a lot on. And I won't go into too many examples of how, like, where this can be used, but it is very useful. Um, and the method that I'll be talking about today is called locator. And so this is built to predict geographic location using genetic data. So briefly, I'll describe how this particular network works. So we start with individual genotypes and genotype locations. This is our training data. These genotypes get shown to the network, and the network predicts a X and Y location for a given genotype. Um, like I described earlier, we compare the true location to this, excuse me, predicted location. And we update the network via this loss function, which is defined as the distance between true and predicted location. And our goal is to minimize that. So get those two get the true and predicted location as close together as possible. And once the network has, satisfied that, we can feed it a new genotype and it will predict us a new location for that genotype. And this network works really well on simulated data. So here I'm just showing you true and predicted locations connected with little lines as dispersal distance increases across the x-axis. Obviously, it performs better when dispersal distance is smaller and there's more population structure to be looked at. And here I'm showing you just error along the y-axis with different numbers of training samples and SNPs, similar idea. So this network is working well on our simulated test cases. And when we take it out for a spin on empirical data, we also get really accurate predictions. So for the human data set, we get predictions within about 10 kilometers on average. However, we notice some really big outliers in terms of errors. So here, again, these predictions are connected via lines to their true location. And so we were curious what is going on when we have these really big errors in our predictions. We thought our network was working well. So to illustrate what we think might be happening with the errors, I'll zoom in on this individual from the human data set that had the largest prediction errors. And this individual is someone with Chibo ancestry, which in fact means that prior to about the 18th century, their ancestors lived in Eastern China where they're being predicted to be from, rather than where they currently live now. So when it comes to humans and we have a good idea of where we've been in the past, we see that our prediction errors are actually reflecting something about ancestry and biology. Considering anopheles a little bit closer to what we'll be thinking about today, we again see prediction outliers in our errors, particularly in West Africa. And this kind of aligns with what we know about population structure, where West Africa tends to have less structured populations overall. So we're thinking that when we get these big prediction errors, they seem to be reflecting something about biology and spatial processes. And so we can, begin to try to start interpreting what's going on. Further, when we took this out, tried the same network on the PF7K data set as well, and kind of squinted at it, we saw that there were kind of similar prediction errors between Anopheles and Plasmodium. And we're wondering whether or not that is actually the case, and whether or not we can interpret these errors as something from biology. Um, so with that, I'll go into a little bit about what we're predicting with these Anopheles and Plasmodium data sets and whether or not we can get at the similarity between our prediction errors. So the Anopheles data set has gotten expanded since then. I also would just like to note for the rest of this talk, I'll just be talking about Anopheles gambiae and Plasmodium falciparum. I probably will end up shorthanding it to Anopheles and Plasmodium, but those are the particular species I will be focusing on. So the Anopheles dataset has expanded. Here I'm showing you the sampling locations in green connected to their mean predicted location. And the size of that blue bubble is showing kind of the error. And we can look at these guys and then again next to our Plasmodium and see that particularly in West Africa, we're seeing similar patterns of error. So little sidebar, how are we predicting location for these data sets in particular? We are training the network on 90% of the data set randomly sampled, and then holding out another random 10% of the data set for prediction. So we have these guys' true locations as well, and move in windows across the genome to predict locations. And then that individual's prediction gets averaged as a centroid of predicted locations across the genome, and we can connect that back to its true location. We do the same thing for Plasmodium, and if we do this iteratively so that we get predictions for everybody, we get these kind of like similar prediction errors. So are these correlated? Can we measure this, quantify this? So to do this, I'm gonna grab a mosquito and pair it with a spatially proximate Plasmodium sample. And to test for correlation, we'll look at the magnitude of these prediction errors, their angle relative to 0. And then finally, using this kind of funky weather metric, actually, to measure the correlation between the vectors of the errors themselves. And to create a null distribution to compare these correlations to, we'll run the same tests just with randomly paired samples with no regard to spatial stuff. Um, so if we look at the correlations between spatially paired samples for vectors, angles, and magnitude, we see that they're all well above zero. And comparing to our kind of like spatially random null distribution, those correlations lie well outside what we would expect based on just random pairing. So it seems like there is something actually truly there, not just something that we're squinting at, but there is some level of actual correlation between these two species prediction errors. And so what's happening here, what we were first suspecting is that we know these mosquitoes are moving across the landscape, and you know, Plasmodium is an obligate parasite, so at some point in its life cycle, it's going to be hanging out inside these mosquitoes. If these mosquitoes are moving at the same time, that parasite's going to move right along with them. And when these guys end up in a new location, we'll end up getting similar prediction errors since they're both more genetically similar to those populations from which they originated in another part of the landscape. And so what we end up getting is kind of this signal of similar prediction error. However, when we were also squinting at this, we started to worry about whether or not this was actually what we were interpreting or whether or not we were getting biased inference. So you'll notice that a lot of our samples also come from this region where we're getting a lot of our prediction errors. So are these errors from this biological interpretation that we're trying to assume is happening, or is it just bias? And so what do I mean by bias here in particular? Um, so I'll bring us back to our supervised machine learning toy example. And recall that, you know, this network is trying to learn something about the training data to associate it with labels. Um, in theory, what we'd like to think is that there's some kind of latent variable in this training data that the network is going to be able to pick up on in order to label the data. So that when we give it new data that has this latent information in it, that's what the network is quote unquote seeing to predict the labels. Um, However, this is not always how machine learning works in practice. Um, and so, as an example that I like to use, Amazon's AI resume screener tended to select resumes that belong to men rather than resumes that belong to women because of this same kind of biased issue. And so, what's going on here is that the distribution of the training data is not representing the true distribution of the data we'd like to predict on. So for example, if you train a network based on a bunch of resumes that belong to men and say that those resumes are what got you hired, we might be expecting the network to pick up on, you know, your education or your job skills. But what it's really learning is that these resumes are belonging to men. So when we give it new resumes, the network isn't going to perform very well on them because it's not picking up what we actually want the network to be picking up. So back to genetics, what does that mean for our own machine learning? So you'll notice again that the majority of our training data does come from this area. Um, and when we're training Locator, what we're wanting to think is that the network is learning something about the relationship between genotypes in space, something that can tell us about spatial ancestry. However, if our samples are all from the same place, we're worried that what the network is actually learning is that those samples are from that location. And it would just be easiest to predict, you know, to where all of the other training samples have come from. So in that sense, we end up with predictions that kind of project towards the middle of the densely sampled area, rather than maybe reflect the biological interpretation that we're wanting to look at. So it's kind of the big question here is whether or not these errors are interpretable through biology or if we're just getting biased results. Um, to first investigate this, I started with spatial simulation. So I very roughly approximated the distribution of Anopheles gambiae across their range. This is not a great accurate simulation in terms of biology, but what the key thing that's happening here is that they are randomly dispersing. So the mosquitoes in the west part of the landscape are moving just as much as the east part of the landscape, and we shouldn't expect kind of any level of prediction error that's greater in one part of the landscape than the other. Um, so if I sample this the simulation with the same distribution as the real data set, it'll serve as kind of a good, I guess, sanity check as to whether or not this sampling distribution is causing big prediction errors in one part of the landscape. So showing you our empirical predictions again, and now on the right, the simulated ones. So we can see that when we have just truly random dispersal across the whole landscape, we get a lot more accurate predictions than what we see with the empirical data, particularly in West Africa. And we can also see that the distribution of prediction errors is more kind of like normally distributed rather than bimodal. So that makes us feel a little bit more confident about the fact that the prediction errors that we're getting are not just due to the distribution of the training data. However, it is true that these sampling locations are uneven across the species range. And in fact, that's true of a lot of biological data sets. Here's just another example of gray wolves. It's hard to sample everywhere that a species can be. And if we want to interpret the machine learning errors that we're getting as being due to biology rather than, biased inference, we need to understand when the predictions are biased by spatial sampling and maybe how we can account for that. So with that, I will begin talking about how we can start thinking about spatially biased data sets and maybe dealing with them within this network. So again, to investigate how spatially biased data sets affect our predictions just generically, I'll again use spatial simulation. And I have these, again, using randomly dispersing populations. They kind of run across the landscape like that, mate with spatially proximate guys, disperse, go on. And these are going to be what I'm using to test locators, I guess stress test locator really. Um, to generate kind of artificial spatial bias in our training data sets, I'm taking these simulations and sampling them with basically an increasing probability of being from the right half of the landscape. So creating bias in these data sets along the x-axis. These training data sets are then given to Locator. So we train Locator on genotypes and locations from these spatially biased data sets. And then once the network is trained up, use it to predict locations for new genotypes. And to investigate whether or not the spatial distribution of samples is impacting our predictions, we'll decompose these samples or these predictions rather, into kind of two axes of error, the error along the x-axis and the error along the y-axis, illustrated with my lovely little triangle there. And the idea here is that if we predict across a bunch of locations that are evenly distributed across the landscape, if we've trained on biased data, then we would expect that our predicted locations tend to be to the right of where our true sample locations are. So the metric that we're looking at is essentially whether or not the average x-axis error is above 0, so as more to the right than these samples truly are. So along this axis, the x-axis, now I'm showing you that training set bias, and along the y-axis, the x-axis error, I guess. That's a little confusing, but essentially our metric of prediction bias. Each of these blocks represents an increasing dispersal rate. Error gets bigger as dispersal increases. And we can see that as training set bias increases, we also tend to get more positive values in terms of x-axis error, which is, you know, exactly what we'd expect. If you train a network on a really biased set of data, you're not going to get accurate predictions for a more generic set of samples. And this is definitely most pronounced at higher biases and with higher dispersal rates as well. Um, if we consider error along just the y-axis of predictions or just the generic magnitude of the errors, we don't see the same impact of training set bias, which is really confirming that it's the x-axis bias that is contributing to these prediction errors. So we have spatial bias and it leads to biased predictions, which is something that is kind of to be expected, but also not ideal for our goals in interpreting locators errors. Because we want to think about these errors in terms of biology, not in terms of just our sampling scheme. So can we help this in any way? Can we reduce this bias? First thing that we tried to account for spatially biased data was just downsampling. So just this is a very extreme example, but just, you know, picking out samples to create kind of a more even distribution across the range than what we truly have. This does not work very well. So here I'm showing you in dark blue, just the, I guess it says unweighted, but let's just go with the generically biased training sets. And in pink, we're seeing the x-axis error for downsampled. So, you know, maybe we're reducing the bias, but our predictions are all over the place. And when we're removing data from the data set, we essentially are just getting bigger errors. So downsampling is maybe not the move when we don't have enough data. And it's important to kind of use every sample that we have. Otherwise, we're losing our prediction accuracy at all. So what can we do that's not downsampling? Um, to introduce, so we're going to try weighting the samples. So recall that during training, the network is trying to minimize loss so that distance between true and predicted location. And so what we're doing with sample weighting is essentially giving each sample that we're training on a relative importance and using that to kind of augment the loss, um, such that samples that are in really dense areas are kind of given less attention by the network. And it's more important to get accurate predictions for samples that are in less dense areas. And so by doing this, we can, you know, still take advantage of every sample that we have in the data set, while also accounting to some extent for there being more dense sampling in some locations. And yeah, essentially getting the network to pay even attention to the whole landscape regardless of sample density. Um, when we're creating these sample weights, we have two parameters that we're thinking about. The first is bandwidth, so just kind of like how large the kernel is that we're using to assign sample weights. So smaller bandwidth, you're only considering your very close neighbors, and then as bandwidth gets larger, this kind of like density assignment gets kind of more smeared. Um, the weights assigned to this density estimate are inversely proportional to the density, obviously. And then we also are going to modify this weight parameter using what we were calling lambda, which essentially kind of scales the difference between the smallest and largest sample weights. So at a smaller lambda, sample weights will all be numerically pretty similar to each other. And as lambda gets large, you end up with really big differences between your most important samples and your least important samples. Um, Obviously, adding in a bunch more parameters to tune is kind of hard to deal with. And so to identify the best like bandwidth and lambda parameters for our sample weighting scheme, we performed a grid search across a bunch of them. And our goal with the grid search was actually to minimize overall prediction error rather than with an eye towards the biased axis, because in a vacuum, you don't know which way your bias is actually going. Here we do, but when you're using real data, your only metric is really how big your errors are. So showing you now the very big grid search across bandwidth and lambda values. And it's very easy to make prediction errors very bad with this scheme. However, we also were able to reduce prediction errors beyond just the unweighted versions. And the lambda bandwidth parameter that ended up reducing prediction error the best relative to unweighted data was actually this very small lambda, very small bandwidth. So the sample weighting scheme for this data ends up looking something like this. So we have subtle weighting and not a lot of difference between the most important samples and the least important samples. However, if we use this to modify loss, we actually do end up kind of not completely eliminating, but reducing the bias in our predictions, which is exciting and a good way to, reassure ourselves that, if we are trying to interpret locators' predictions through biology, there we can reassure ourselves using these sample weights to show that maybe there's less bias in the predictions. Works a lot better than downsampling. We do still see overall kind of increased variance in our predictions, which comes down to whether or not you can be very good at predicting a small set of things or pretty good at predicting a large set of things. Bias variance trade-off is always out to get us. But we were excited about this and particularly interested in whether or not, applying these sample weights would tell us more about what's going on with the Anopheles plasmodium system that we're trying to say something's happening biology-wise. So with that, I'll just come back to Anopheles and plasmodium again. So we're looking at these common patterns of prediction error between host and parasite, and are concerned about, the prediction bias with our samples. Are these errors due to whether or not these guys are moving together, or are our predictions biased towards the center of the landscape? So we tried out our sample weights on our test data sets, and using kind of like that same grid search across lambda and bandwidth parameters, we made sample weights for the Anopheles and plasmodium data sets, trained up locator, and did the exact same thing. And here I'm showing you the results from that. So the error distributions down here are shifted slightly, and we get slightly different predictions between the unweighted and weighted runs. But overall, we still see really common patterns of error, especially in West Africa. And when we do our same like formal correlation tests, we do see reduced correlation. So obviously there was some bias happening, but our correlations between vectors, angles, and magnitudes of errors are still well outside what we would expect based on just our randomly paired samples. So what we think is happening here when we're seeing these correlations is that we're detecting common patterns of movement between host and parasite, or vector and parasite, I guess is more formally correct. So when these mosquitoes are moving around, sometimes plasmodium is hanging out inside of them, and they end up being able to travel to similar places, and we get prediction errors that are similar between them. So because they're traveling together, they'll both be more genetically similar to that point of origin, and will be predicted to be back there. And so this is kind of cool that we're able to find this with machine learning, and particularly not only just correct predictions, but when it's messing up. So yeah, interpretable errors are kind of a cool thing to be able to look at with the system. Um, so overall, there are different reasons that we can be getting prediction errors with this method, and spatial bias in the training data is a very big problem that we have to think about when we're using this method. However, we have developed methods to reduce the impact of training set bias on those predictions, and demonstrated that our predictions can still be interpretable through biology. Um, and when we kind of sum this all up on our Anopheles and Plasmodium, it really does look like these prediction errors are reflecting some sort of paired geographic population structure between these two species. Some kind of like follow-up thoughts for what to think about next with this stuff is how epidemiologically relevant genotypes are moving across the landscape. So, if vector and parasite are moving together as it seems they are, where are they going and what are they taking with them? Um, Another kind of axis of investigation is how human movement contributes to these paired migration patterns. So the errors that we're seeing also correspond pretty well with axes of human movement. Obviously, we're not moving on the same generational time scale as mosquitoes and malaria, but incorporating human movement data into this analysis is something that I'm excited to do. And then also looking into whether or not we see these paired movements in other parts of the world. So with that, I guess I kind of sped through that. But I just want to thank my lab, especially Andy and Peter, Scott and Nate, and then CJ is the one who concocted Locator to begin with. And also thanks to all of you for listening. And thank you so much for having me. And yeah, I guess I can take questions.
Thank you so much, Clara. This was amazing, as expected and anticipated. So thank you so much. We've now reached time for Q&A as well as discussion. So kindly note that you can raise your hand if you want to speak, and we'll allow you to ask your question, or you can type your question in the Q&A or chat box. And I see we already have a question from Chun Lee, who says, "Hey, Clara, could you provide a bit more information on that metric that comes from the weather analysis that you mentioned to measure the alignment of two vectors? Is it dot product based?" That's from Chun. Clara?
Great question. Thank you. I believe it is dot product based. I will I have to find you the paper for that. I do believe it is dot product based. We dug up a old like GIS paper to find this metric, and uh that is about the extent I know statistics wise about it. However, I can follow up with you on that. And also, if you have any other thoughts about computing correlations between vectors, that would be awesome.
Okay, thanks, Clara. More questions are popping in. The second one is from Marie-Lou Baudet, who says, "Thanks for the beautiful talk, Clara. It is still an open question how far mosquitoes disperse and how heterogeneous dispersal is within populations. Can you say anything about what range of dispersal parameters you used in your simulations and potentially how that compares to empirical data?" That's from Marie-Lou.
Awesome. Thanks, Mary Lou. So in regards to the simulations that I did that to approximate the mosquitoes, I based their dispersal kernel on I believe Chris Smith's estimate of per generation dispersal, roughly like 10 kilometers per generation. However, it was just like a Gaussian dispersal kernel, and I know that particularly with Anopheles, there's a lot of discussion as to kind of like long-tailed dispersal. Some individuals tend to move a lot farther than the rest of the population. So I think that in the mosquito simulations, dispersal, particularly in West Africa, in reality is going to be a lot larger than what I was simulating, which in part contributes to those large prediction errors that we're seeing relative to East Africa. However, yeah, it could be refined in the randomly dispersing simulations that I was using to kind of stress test locator. Dispersal ranges were between, I guess, maxed out at about 1 unit, so about 1/25th of the whole range of the landscape. So I would guess probably relative to the whole range of mosquitoes, a fair bit larger than a mosquito will be dispersing across the range that we're looking at. Hopefully that answers your question.
Thanks, Clara. We have one more question in the Q&A, but just to repeat to the audience, you are allowed to raise your hand, and we'll allow you to unmute and speak. The other question is from Jacob Amalgro Garcia, who says, "Great talk, Clara. The question is, how big are the sliding windows of genotypes you feed the neural network?"
So different sizes for mosquitoes and Plasmodium. For the mosquitoes, I want to say 100,000 base pairs, and then slightly smaller for Plasmodium. The idea with the sliding windows is to, oh, also they are non-overlapping windows, just to clarify. But the idea is to capture essentially enough SNPs to get an accurate prediction. Roughly, I think 10,000 SNPs is what we're shooting for in a window. And beyond that, you end up with diminishing returns and kind of muddy up different portions of genetic ancestry. But you can also, the other way to run this network is feeding it the whole genome, and it will essentially randomly pick SNPs to learn on.
Yeah, thank you so much, Clara. You might have answered this in your talk, but for me, my question that I jotted down was, are the correlations or the errors between parasite and host from biology or bias or both? And can you confidently, because I mean, obviously you went through a great length of explaining, but I just, in simple terms, is it biology or is it bias?
I want to say it's biology. I think that it was, let's see, let me pick back through this now. I think the thing that made me most confident that it was biology was seeing these sample weights like not really make any impact, especially when we at least convinced ourselves that they were able to reduce prediction bias in our test cases. Seeing these large prediction errors, like despite the sample weighting, despite our kind of like simulated null distributions, made me feel confident that these are kind of like biological errors that we're seeing, that reflecting kind of like more homogeneous population structure. As to whether or not it is truly like I illustrated with this one mosquito dragging that one parasite over, I would say I maybe less the case, but the shared like patterns of population structure, I think, is the biology that we're seeing. If that answers the.
It does. It does. Thanks, Clara. It does answer the question. I appreciate it. We still have some time for questions and comments. So please do type in the Q&A or raise your hand. Marilu, go ahead and unmute yourself.
Thanks. So here you talk a lot about how mosquitoes and parasites can have shared patterns and like how parasites might be moved by mosquitoes. But parasites might also, of course, be moved by parasites might also be moved by humans. Do you have any ideas how, like what would you need to also be able to incorporate that in this analysis, or do you have any ideas how to go about that?
I've been thinking about that a lot actually, because obviously we can't, do the same thing with human genotypes and expect the same results because we move on very different scales generationally than these other two players. Some data I've been trying to hunt down kind of like more like human population movement data. There's like migration statistics. I've like road movements, like cell phone data is something that I've been even looking at. Um, it's I have a lot of like ideas of what would be interesting in terms of, what can tell us about where humans are moving in this landscape. But it kind of becomes more of a question of who has that information and whether or not I'm allowed to see it.
Yeah.
But I, but yeah, I think that something like, yeah, I think even like, yeah, cell phone movement, I think is one of the big ones that I think would be really interesting to look at. Um, or something kind of analogous to that, like road usage metrics. I'm gonna have to like talk to some city planners or the social scientists that we'll have at GEM next time. Um,
Very cool. I'm looking forward to it.
thanks.
Thanks for your question, Marilu. We still have time, as I said. So please do go ahead and raise your hand or type in the chat box. We have about 10 minutes to go. Okay, Chun, go ahead.
Hey Clara, it's Chian. Yeah, good to see you again. And thanks for your talk. Yeah, I was talking to someone at Gem, and they explained to me, for things like RNA-seq, you typically sort of cluster on data, and then you like zoom in on a subset of that overall sort of mega cluster, and then pick out a mini cluster, and then sort of cluster on that again. And it just sort of reminded me maybe you could sort of resolve that cluster in West Africa a little bit better if maybe you just ran that by itself and potentially with just more samples. And I noticed sort of the area sort of it looks kind of radially distributed. So I wonder if I don't know, sort of tweaking your model to instead using like Cartesian longitude latitude using some sort of polar system might be kind of interesting, but not sure if that'll do anything. But yeah,
A
that
polar system would be very interesting. I
yeah, I think it'd
haven't
be
even
quite interesting. Yeah.
the oh yeah, thinking about that would be very interesting. We have tried to pick out I've ran locator on just the West African samples and you do get similar error, but again, they are kind of projecting in a similar fashion. That's really the radial idea is interesting. Sorry, I forgot the other part of your question.
And no,
I
it
was
wasn't really a question,
distracted by
more of
your
a comment.
question. But yeah, that would be a good idea. We have tried to, I think, dig into the like population structure in a couple other ways. Like one of our reviewers asked us to do like correlation between like principal components, which ended up in the supplement. And I didn't talk about it here, but you see like same correlation, like something you would expect. But trying something else in terms of prediction to like optimize that area would be a cool approach. We should talk about that. Thank
Totally.
you.
Hey, thanks, Chun, for your question. You can follow Clara on Twitter, add her on LinkedIn, and engage further on the conversation because I think this is really interesting work. And as I said, Clara, your title giving or the way you give titles, because I remember your poster at GEM as well. I'm impressed.
Thank you.
We still have a bit of time for questions. I'm going to give it a few more seconds before I close. So I'm going to go once, or twice, 3 times. Clara, any last words from your side?
Thanks y'all so much. Thanks for
Okay.
listening to me.
This was really fascinating and engaging, Clara. And your paper is really fascinating as well. So thank you so much for your time. Said that you had to be back in the US and you couldn't do it physically here, but I'm glad you could join us online. The recording is going to be
Y'all.
up on Malaria Gen YouTube channel later today. So please do check out our YouTube channel. Do follow us on Malaria Gen GSU, LinkedIn and Twitter, and join our newsletter so that you don't miss announcements about our future journal clubs. For the next one, in October, we are going to have Andrew Barmer speaking about the parasites paper that they just released a few weeks ago. So please do check that out on our social media. We'll be advertising it. And there are comments coming in praising you, Clara. Ishla is saying, great talk, Clara, and thanks for getting up so early to chat to us. And yeah, I just want to say we do appreciate your time, Clara, and hope to see you at the next gem where we're going to invite social scientists as well. And to all of you, thank you so much for joining us today. See you next month for the Future Journal Club. Vicky saying, excellent talk, Clara. Thanks so much. Thank you, everyone, for joining us. We'll see you next month. Have a good day.
Well.